Hello. Last lecture we talked about gastrulation and the stage which follow blastulation, and the main aim of the gastrular stage is to form two layers embryo. One layer will give rise to endoderm and mesodermal organ, and the other one will give rise to the ectodermal organ. You can see the examples of each layer and what organ it will give in future from the endoderm and mesoderm and ectoderm. Going through the process of formation of gastrulation, there is like 5 different mechanisms that gastrulation can use to do the process. The first one is invagination, which is the infolding of a region of cells to the inward, as the figure shows. The second one is the involution, in which there is interning or inward movement of an expanding outer layer so that they spread over the internal surface of the remaining external cells. The next method is ingression. It's a migration of individual cells from surface layer into the interior of the embryo. The third one is the delamination. Basically, it is a splitting of one cellular sheet into two parallel sheets. And the last one is epipoly. It is the movement of epithelial sheets that is spread as a unit rather than individually. And that's differentiated from ingression. This slide is an assignment. You need to answer all these questions and submit it to me by email. You can use internet resources, maximum 1-page A4 for the answers of the five questions. We'll talk now about cell specification, a process that happens once a gastrulation is forming, and it's the generalization of a specialized cell type, and this one as well is called differentiation. This differentiation is the last stage of a series of events that make the cell particularly committed to give certain fate. And this table, just for knowledge, to know how the type of cell and its differentiated product and its specialized function. For the commitment of fate of cells, the process of commitment can be differentiated into two stages. The first stage is a specification, in which the cell is capable of differentiating autonomously by itself when placed into a petri dish or a test tube. That's to say, in a neutral environment. And this type of cell commitment is reversible. While the second stage is determination, when the cell is capable of differentiating still autonomously, even when placed in another region of the embryo, that's mean non-neutral environment, and in this case cell commitment is irreversible. There are 3 major strategies of commitment that can embryo do, either autonomous or conditional or syncytial specification. For autonomous specification, and this occurs mostly in invertebrate, the blastomeres, as we said before, inherit a set of transcription factors that exist in the egg cytoplasm. For the conditional specification that happen in vertebrate and the few invertebrate, the ability of cells to achieve their fates by interaction with neighboring cells. So that means the cell, to reach its fate, it needs interaction from the neighboring cells, which is totally different from the autonomous. The blastomere's fate depends on the position in embryo and is specified by paracrine factors secreted by neighbor cells. So you can see here that if we have these blastula, and you remove some cells from the normal back cells of the blastula and transplant it into another region in another blastula, you will see that the cells which originally will form the back of embryo will form the belly tissue. That means that the fate of the cell have been changed due to its location. In another example, you can see that they use a glass needle to remove the cell at all and making a hole in the blastula, but still there is a normal development and there is a compensation of the missing part. That means that cells uses the new position to compensate or condition the specification. Dirichlet was the first scientist. He's a German scientist who provided the first experimentally observable evidence of conditional. He said that interactions between cells determine their fate rather some cytoplasmic factor. Still, the cells have cytoplasmic factor, which is the transcription factors or morphogenic determinants, but the interaction and the paracrine secretion between the neighbor cells enables this conditional specification. In this example, he took the 4-cell stage of an invertebrate animal. He removed the fertilization envelope and separated the 4 plasmid stage into separate 4 cells, and left these 4 cells to continue development. He found that 4 same larvae have been produced. The only difference that the size is different, and they look the same as the original one which developed from the 4 blastomere stage. This means that every cell behaved as a single cell, as a single embryo. Okay, the only difference between the normal Bluetooth larva and the 4 other Bluetooth larvae is the size. Again, Dirichlet continued to explain that he took the 8 blastomere stage and removed the first top 4 cells, and the other separates the upper animal pole cells and lower vegetable pole cells. He found that the lower one developed into Bluetooth larvae that are normally half the size, while the upper half developed the part of the animal which will give to the animals the cilia that help these larvae to move. In another example of the same experiment, he separated in another half in which each half will have 2 cells of the animal pole and 2 cells of the vegetable pole. Both of them develop into Bluetooth larvae. That means that the neighboring cells controlled the 2 halves to be the same, while the other cells there is since in the first part, the first figure, you have the animal pole, all the cells have the same cell type, so each one each half in the first figure give different embryos, while in the other one, since there is interaction between the animal pole cell and the vegetable pole cells, they give whole Bluetooth larva, but it's half the size of normal. So we'll talk about how the morphogen gradients and cell specification. Again, morphogen gradients, it is the biochemical molecules that can be transcription factors that is inherited from the mother ovum of mother egg cytoplasm. These are transcription factors produced within the cells or paracrine factors that affect neighbor cells. If the cells are uncommitment cells, this is exposed to high concentration of the morphogen, are specified as one cell type. When this morphogen concentration drops below a certain threshold, the cells are determined to another fate. When the concentration falls even lower, there is a third fate can be specified. So in this simple example, if we consider if we consider a cell color which is blue, white, and red, and we consider there is a gradient of the morphogenic factors starting from the source high concentration into the sink lower concentration, so you can see that when the source have a concentration, when it decreases until it reaches the threshold of blue, the color change into white, and when it reaches a threshold of white, the color change into red. That means the color change it's like a reflection of the concentration of the gradient. In another example, if you look to the flag, the France and the US flag, if you take switch parts of the flags of the France between the France and USA flag, you will find that the region that it's transferred from the American flag to France flag developed into the same position stars as it's on the original flag. That's mean it as if it took the memory of the region of the same flag, while the other one, when transferred back, it developed the same part of the original France flag. So these reciprocal transplants develop according to their final position in the donor flag. Okay. The third type of specification is happening is insects is called syncytial specification. And what's meaning of syncytial? Syncytial, that means you have like a bag that's full of cytoplasm and there is a lot of nuclei with no cell boundaries. So it's like nuclei is swimming into cytoplasm. So the transcription factors or the morphogen are all over the cytoplasm and surrounding all the nuclei. So if you can see in the larva of the insect in the first figure, you can mainly have two morphogenic transcription factor, which is the bicoid and caudal. And the higher concentration of the bicoid is in the anterior, while the higher concentration of the caudal is in the posterior. And there is a gradient decrease as long as we move into the middle. So in this case, the concentration and the ratio of the two transcription factor will determine the fate and the specification of the cells. And you can see that in the anterior part it's more bicoid and in the posterior part is more bicaudal. So that's why the bicoid is red and the caudal is blue. So that's why in the second figure you'll see that the nuclei turn to be red because it's affected by the bicoid, while the other one turned to be blue because it's affected by the caudal. And you will have in the middle, there is a mix of the true transcription factor which will develop into the other organs. That's mean in bicoid responsible for the formation of the head region, and the caudal is responsible for the formation of tail. Once the concentration fail in the middle, because the gradient is the minimum in the middle, the other organ are formed, either thorax and abdomen. Axis formation. Cell fates are specified by cell-cell signaling and the asymmetric distribution patterning of the transcription factor. These TFs activate or repress transcription of specific genes that acquires them. This asymmetrical distribution pattern of the molecules begin during cleavage, because as we said before, the cleavage distribute this transcription factor these distribute along the blastomeres. So this asymmetric distribution of patterning molecule begin during cleavage and generally follow one of the three mechanisms. The molecules are bound to the egg cytoskeleton and is passively acquired by the cell to obtain the cytoplasm. The molecules are actively transported along cytoskeleton to each particular cell. The molecules become associated with a specific centrosome and follow that centrosome into the two mitotic sister cells. So basically, this is the three methods of how these morphogenetic molecules or patterning molecule or transcription factors are recruited into the developing blastomere during the process of cell division. Once this asymmetry has been established, one cell can specify a neighboring cell by paracrine interaction. So embryo, by the end, for the axis formation, had developed into three crucial axes: anterior-posterior axis, dorsoventral axis, and right-left axis. And you can see here from the figure how these axes are illustrated on the figure. Four invertebrate groups, sea urchin, snail, ascidians, and C. elegans, have been important model system for developmental biologists. Why? Because they easily maintained in the laboratory and represent a wide variety of cleavage types, pattern of gastrulation, and ways of specifying axis and cell fate. Despite their differences, the embryo of these four invertebrate groups are categorized as type 1 embryogenesis. Why type 1? Because there is immediate activation of zygotic genes, there are rapid specification of blastomeres, and the number of cells are few at the start of gastrulation. Early development in amphibian axis formation. Amphibian embryos, including frog and salamander, is the organism of choice for experimental embryology because they have large cells and have rapid development. If you look to the egg of the amphibian, the characteristic, its type is mesolysisal egg. So you see that yolk is concentrated in the vegetal hemispheres, and this delays cleavage. Thus, the first cleavage begins at the animal pole, slowly extend down into vegetal pole. So you can see that the molecule that specifies the dorsal fate are located ventrically, the one with the light blue. The sperm enters from the left side, as you can see, and the cytoplasm at the edge of the cell is called cortical cytoplasm. Once there is a fertilization, as we said before, there is a cortical rotation. This cortical rotation leads to the merging of the cortical cytoplasm of the ventral part with the cortical cytoplasm of the animal part. So the merge leads to the movement of the molecule, the blue molecules, into the dorsal side, and the formation of region is called gray crescent. So gastrulation begins and starts at that location. So this gray crescent established the bilateral symmetry of the frog even before the first cleavage event, because the first cleavage event bisects. So the first cleavage bisect this gray crescent. So the site of sperm entry will become ventral to the region of the embryo, the belly, as in the figure, and the gray crescent will become the dorsal region of the frog as a back. So you can see it's another illustration of how after the entry of the sperm there is a rotation of the cortical region of the cytoplasm to appear the gray crescent. Once the gray crescent is formed, the first cleavage starts, and it must start and bisect the gray crescent, defining the dorsal and ventral axis. That's why gray crescent establish the bilateral symmetry of the frog. And hence the larva is defined from the early first cleavage to have the axis that we have been said before. The effect of why the gray crescent is so important for bilateral symmetry. Experiments have shown that if we have an embryo and after the first cleavage the two blastomere are separated from each other, it will give two normal developed embryo. That's mean and equal sized. That's mean it's a kind of that if the gray crescent is not important for bilateral symmetry, the two embryos arise will not be the same. Again, if we separate the two embryos when only one of the two plasmere receive the entire, so we assume that if the first cleavage does not pass into the gray crescent and we separated the two blastomeres, we'll have a normal embryo which received the gray crescent, and the other will just a belly piece of cells that is not differentiated. If we look again to the cleavage, you'll find that the first cleavage followed by second, third, fourth, and go on until a ball of cells is formed. And due to the concentration of the yolk at the vegetal part, there is retardation in the cleavage furrow, leads to formation of two types of blastomeres, and the cavity formed is shifted to the animal pole and is called blastocele. You can see again that the first cleavage passes through the gray crescent. Again, the cleavage furrow, of course, are numbered in orders. The second cleavage is retarded. The third cleavage is displaced toward the animal pole because the yolk is concentrated into the vegetal pole. That's why the vegetal hemisphere contain larger and fewer blastomere than the animal hemispheres. And the last one is like H, figure H represents the mid-gastrulate stage embryo. It's a transition stage between the in the gastrulate stage. If we look to real image scanning electron microscope of the cleavage, you can see how the first cleavage appear. In the below figure, it's a scanning, so you can see the first and second cleavage and the cleavage furrow, and around like the 8 to 16 blastomere stage, figure C. It's again the cleavage at the right angle. The first and second are right angle to each other. The third cleavage is equatorial and displaced, and it divides the blastomeres into micromeres, which is upper, and macromeres, which is down at the vegetal blastomeres. And this type is unequal radial holoplastic cleavage. As cleavage progresses, two populations of blastomeres arise, rapidly dividing animal micromeres and slowly dividing vegetal macromeres, and this we call it yolk-laden macromeres because they have all the yolk of the embryo. From 16 to 64 cell stage, we call it morula stage, while at the 128 cell stage, the blastocele is forming and it's fully formed blastula. If you continue and see the importance of a protein that is important in adhesion, which is called the EP cadherin, while blastomeres are dividing, EP cadherin is one of the numerous cell adhesion molecules that keep the blastomere together. So this messenger RNA for that protein is already supplied by the oocyte cytoplasm. So if this message is destroyed, in other means, if the EP cadherin is not made, the adhesion between the blastomere is dramatically reduced, resulting in the destruction of the blastocele. So you can see here that control embryo, that's mean the embryo is normal. You can see the blastula with the blastomeres, while in this embryo which have the EP cadherin is depleted, you will find a massive cell with undifferentiated blastocele and no orientation and no differentiation.